Okay, thank you for the introduction. Welcome back from lunch. Okay, so yeah, first of all, it is my pleasure to join this conference and see CHAMP1 families and scientists again. Then I'd like to thank Jeff, Daniel, and the other organizers for their hard work in putting in such a wonderful conference. And then it is amazing that this CHAMP1 research community is getting bigger and bigger and has now joined with the Kozo research community thanks to the efforts of the CHAMP1 Research Foundation as well as the White Sutton Syndrome Foundation. Okay. Sorry. All right. So I'm from Tohoku University, which is in city of Sendai in the northern part of Japan. And this is the Institute of Development Aging and Cancer, where I've been working as the director. And these are my lab members. So we published our first paper on CHAMP1 in 2011 and have been working on these genes ever since. And these are human chromosomes. We have 23 pairs of chromosomes, and our 19,000 genes are distributed over this chromosome. And the CHAMP1 gene is located at the end of chromosome 13. And from this CHAMP1 gene, CHAMP1 protein is produced. CHAMP1 is short for chromosome alignment maintaining phosphoprotein 1, which we designated because of its role in chromosome segregation. This is a diagram of CHAMP1, and the proteins are made up of amino acid, and the CHAMP1 is 812 amino acid long. And proteins are, can be divided into domains which have different structures and functions. And CHAMP1 has zinc finger domain at both ends, shown in blue, which provides a structure for binding to DNA, RNA, and proteins. And this middle region has several repeat motifs shown in different colors, and this region doesn't form a discrete structure known as intrinsically disordered region. And this structure predicts that CHAMP1 has globular domain at both ends connected by a flexible region. And this was confirmed by this movie, which was taken using high-speed atomic force microscopy. And here you can see two globular domains are connected by a flexible region. Since 2015, CHAMP1 mutations in individuals with neurodevelopmental disorders have been reported in many papers. And here, three types of mutations: missense mutations, nonsense mutations, and frame shift mutations are shown in blue, red, and green, respectively. And as you can see, most of the mutations are nonsense or frame shift mutations, and in some cases, the whole CHAMP1 gene is lost by deletion. However, the CHAMP1 Research Foundation has much more information than these papers, and in the list of the mutations registered with the foundation, now there are many missense mutations, not only limited to the beginning of the sequence, but throughout the CHAMP1 sequence, showing the great contribution of the CHAMP1 Research Foundation for our understanding of the whole picture of the CHAMP1 mutations. So maybe you already know, but let me briefly explain the types of mutation. When messenger RNA is used as a blueprint for a protein, A combination of three nucleotides, as shown here as A, G, C, or T, correspond to one amino acid, which is called the genetic code or codon. And if so, that when mutation occurs, some nucleotides are altered or lost or gained. And if a corresponding amino acid is changed to another amino acid, it is called missense mutation. And then if a corresponding amino acid is changed to stop codon, which stops the elongation of amino acid, it is called nonsense mutations. And if, on the other hand, if there are some nucleotides are lost or gained, it causes a shift in the reading frame, which is called frame shift mutation. And if it occurs, the amino acid sequence is changed completely thereafter, and then the elongation eventually stops by the appearance of stop codons. Therefore, nonsense mutation and the frame shift mutation give rise to the formation of truncated CHAMP1 mutant, collectively known as premature termination codon or PTC mutants. So importantly, it's already shown in the previous talks. POGZ is also mutated in individuals with neurodevelopmental disorders. And this is the diagram of POGZ, which has also zinc finger domain shown in yellow, and bind to CHAMP1 at the tail region. And here the mutations are colored according to whether they are seen in autism spectrum disorder or intellectual disability or other neurodevelopmental disorders, collectively known as white Sutton syndrome. So we've been working on CHAMP1 for more than 10 years and have reported that CHAMP1 has several functions such as proper chromosome segregation, homologous recombination DNA repair, cell survival during cell cycle arrest, and neuronal development. And for homologous recombination DNA repair, Alan has also published great work, and then he will show the progress of the work in the next talk. Interestingly, POGZ was also originally reported in its role in chromosome segregation. And POGZ is also involved in homologous recombination DNA repair, as Alex showed in his talk. And then POGZ also plays a role in neuronal development, as Aline presented in her talk. So these overlapping roles between CHAMP and POGZ highlight the cross relationship between them and the importance to discuss these proteins together, for which the CHAMP Research Foundation and the White Sutton Syndrome Foundation has contributed greatly. Another contribution of the CHAMP Research Foundation is to make biological samples from CHAMP individuals publicly available for CHAMP research. And we have obtained lymphocyte and fibroblasts from CHAMP children from the Korea Institute, and found that these PTC mutants were expressed at expected size. And we investigated whether these lymphocytes from CHAMP children had a defect in homologous recombination. As a readout to assess the homologous recombination activity, we quantify the number of phosphorylated RPA2 foci which localize to DNA double-strand breaks repaired by homologous recombination. And as you can see, phosphorylated RPA2 foci was reduced when CHAMP1 was depleted, revealing the HR defect. And then when we quantify the number of phosphorylated lipase foci in lymphocytes with CHAMP1 PTC mutant. So it was reduced as shown here, suggesting that lymphocytes with CHAMP1 PTC mutant show defect in HL repair. So one of the unresolved questions for the role of CHAMP1 mutation is whether did CHAMP1 mutation just lose their function or perturb the function of wild-type CHAMP1. The former is known as haploinsufficient and the latter is known as dominant negative. So to address this question, we expressed these PTC mutants as well as these two missense mutants in UTOS cells, which are commonly used to study homologous recombination. Then we first address whether these CHAMP1 mutants affect the homologous recombination activity of endogenous CHAMP1. And as you can see, so the number of phosphorylated lipase foci was reduced when CHAMP1 was depleted. In contrast, so these CHAMP1 mutants did not affect the number of phosphorylated lipase foci, suggesting that exogenous CHAMP1 mutants do not perturb the HL function of endogenous CHAMP1. Then next, we depleted endogenous CHAMP1 in these cell lines, leaving only the CHAMP1 mutants. And wild-type CHAMP1 rescued the number of phosphorylated lipase foci. Whereas, these PTC mutants could not rescue the number of phosphorylated lipase foci, suggesting that CHAMP1 PTC mutants are loss of function. In contrast, these CHAMP1 missense mutants rescue the number of phosphorylated lipase foci at the level comparable to wild-type CHAMP1, suggesting that these mutants are functional. So in our in previous slide, so that there was a defect in homologous recombination in lymphocytes from CHAMP1 children, while there was no defect in uterine cells expressing CHAMP1 mutant. We hypothesized that the amount of wild-type CHAMP1 in these cells made the difference. To address this possibility, we generated cells with heterozygous or homozygous depletion of CHAMP1. So in cells with homozygous depletion of CHAMP1, CHAMP1 was totally lost. And in cells with heterozygous CHAMP1 depletion, CHAMP1 was reduced but still present. And as shown here, so the number of phosphorylated lipase foci was reduced not only in cells with homozygous CHAMP1 depletion, but also in cells with heterozygous CHAMP1 depletion, supporting our hypothesis. In summary, these PTC CHAMP1 mutants are loss of function, while these missense mutants are functional regarding homologous recombination activity. Accordingly, cells with these PTC mutants are haploinsufficient in HR activity, as are cells with heterozygous deletion of CHAMP1. However, it was suggested that these PTC mutants play a dominant negative role in neurodevelopmental disorder in CHAMP1 children. So these findings suggest the possibility that this HR deficiency may not account for the cause of a neurodevelopmental disorder in CHAMP1 children. So to address the role of CHAMP1 in neurodevelopmental disorder, we established CHAMP1 knockout mice in which one or both alleles of CHAMP1 genes are deleted. And homozygous CHAMP1 knockout mice died shortly after birth, and neuronal differentiation of the embryonic brain was delayed. And heterozygous CHAMP1 knockout mice showed mild memory defect, altered social interaction, and depression-like behaviors resembling CHAMP1 individuals in some aspects. So a number of papers have been published on the analysis of POGZ knockout mice or mice with POGZ mutations, which are informative in considering the role of CHAMP1. And many of them suggest that POGZ is involved in transcription regulation, which may be associated with neurodevelopmental disorder. And as you know, the gene is transcribed into messenger RNA and then translated into a protein. And then when transcription occurs, a number of proteins, including RNA polymerase II, work together by binding to a proximal region of DNA called the promoter. And in addition, the distal region of DNA, known as the enhancer, also plays a role in transcription, as shown here. And one possibility that CHAMP1 work together with POGZ in this process. Yeah, as Eileen showed in her previous talk. Okay, so when we analyze gene expression in the brains of CHAMP1 knockout mice, we found that genes related to neurotransmitter transport were downregulated. And we've also found that genes related to neurodevelopmental disorder were downregulated, suggesting that CHAMP1 is involved in the transcription of genes. Then we study the genomic distribution of CHAMP1 in mouse embryonic brain by performing cut and tag acid, and found that CHAMP1 is enriched in the enhancer and promoter region as shown here, similar to POGZ. And we also found that the localization of the CHAMP1 and POGZ are highly overlapping. Then we addressed the chromatin structure change in CHAMP1 knockout mice. So it is also overlapping with the previous talk, but so the genomic DNA is wrapped around histones to form a structure called chromatin. And if chromatin is tightly packed, transcription factors cannot access the DNA, thus transcription doesn't occur, which is a condition known as closed chromatin. And to start transcription, so this chromatin DNA need to be regionally free of histones to allow binding of transcription factors, a state known as open chromatin. So to investigate the chromatin structure change in the brain of CHAMP1 knockout mice, we performed ataxic assay and asked Stefano for the analysis. So these graphs show transcription factors whose binding sites are enriched or reduced in open chromatin region in CHAMP1 knockout mice. So this is the last slide. So we found that here, so the chromatin structure at the binding site of transcription factors related to neuronal development were altered in embryonic CHAMP1 knockout brains, including ENN1, which is a transcription factor related to the development of the central nervous system, as shown here, suggesting that CHAMP1 maybe may play a role in the transcription regulation through these transcription factors. So we are now investigating this possibility further in collaboration with Stefano. Okay. All right. Finally, I'd like to thank all the collaborators shown here, including Stefano, and I also would like to thank the CHAMP1 Research Foundation for their continuous support for our study and for organizing such a strong research community. So I'm happy to collaborate in any way and to provide any materials and information I have to promote CHAMP1 research. So I hope and I believe we will see a great advance in the development of the effective treatment for CHAMP1 children as well as POGZ children in the near future. So thank you for your attention, and I'm happy to take any questions.
So thank you very much. Always nice to hear you. And questions?
A question from a virtual audience. Great talk again. It seems to be clear from many studies that CHAMP1 plays a role in HDR. So I have the same question as to Alexandra. What do you see the role of HDR in neural developmental delay, since it does not play a role in post-replicative neurons?
A defect in HCR, you say?
HDR.
HR, homologous recombination. Right, right. And then the question was the relationship between homologous recombination defect and neurodevelopmental delay. Okay.
What do you see the role of HDR in neural developmental delay, since it does not play a role in post-replicative neurons?
Okay, yeah. The answer, the short answer, we don't know at the moment. But in my opinion, so the both, the homologous recombination and also for transcription regulation, so the both, required chromatin structure change. So in this sense, in my opinion, so the homologous recombination do not directly explain the neurodevelopmental disorder, but it may through transcription regulation. But still, the chromatin regulation is common for both homologous recombination repair and transcription regulation. Okay. Hi.
Sorry, maybe I missed it, but at some point you mentioned that the missense mutations show a gain of function, right? In the thing. So can you just clarify what you mean by gain of function? Is it the dominant negative?
So this is from the paper from Dr. Zolino. So she showed that some CHAMP1 missense mutation is gain of function. So show epilepsy, I remember. But yeah, I think it may depend on the type of missense mutation. And now in this audience, now there are many missense mutations are found, and then I heard that some of them are very mild phenotype and functional. So it depends. So that it doesn't mean all the missense mutations are gain of function. Thanks.
I have a more curiosity question, and I think we all see the same, which is that, I mean, we would expect that POGZ or CHAMP binds to HP1 and then that maintains heterochromatin. And therefore, when you remove them, you should actually have more chromatin and upregulation of genes. However, when you do your transcription analysis, and I think it's the same data in ESL, it's the same data in TNBC, is that we see both an upregulation and a downregulation of gene, which suggests a more complex model. Do you have any, I don't know, supputation, ideas why, you don't just get an upregulation of genes, but a more very global changes of the transcription that is up and down regulation?
Thank you for the good question. I myself don't know the answer, but you know, it's not uncommon. So the certain transcription factors sometimes upregulate certain types of genes, and while so the downregulate some kind of gene. So there may be a complex relationship, and then we also know that so there are many binding sites for CHAMP1 throughout the genome, but only a subset of genes so that are differentially regulated by RNA seq analysis. So it's not just one by one relationship, but there may be other factors, and there may very complex so the relationship. But so that as far as I understand, it's not only for CHAMP and POC, but other genes so that mutated in neurodevelopmental disorder are more or less in similar situation.
Any other question? Alan?